Hello, students. So, we have started the lesson that is enhancement of food production, and this is enhancement of food production part 6. We have discussed various animals and insects which are helpful in the last lecture in the part of animal husbandry and its importance, the life cycle, and each and everything. So today we are going to discuss the microbes in human welfare. Okay, so let's have a look how these microorganisms they are helpful for the or they are how they are beneficial to the human beings. So, microbes in the human welfare. Now, biotechnology is the applications of scientific and engineering principles of for the processing of materials by using biological agents to provide goods and services to humans or for human welfare. Now, when you discuss about microorganisms, the science, the branch of science which studies or which applies the basic knowledge, okay, so that implementation is done by this branch of science that is called as biotechnology. Biotechnology is very important as far as microorganisms and human welfare is concerned. So there are variety of microorganisms like algae, fungi. So these are various microorganisms which we use. Okay, so these are the types like algae, fungi, bacteria, again viruses, protozoa, nematodes, etc. So these are number of microorganisms which are used in various areas for the uh, benefits of human, okay, of humans, and the products that exhibit beneficial activities which are used for welfare of humans is regard to where they are used. Now they are used in the food, health, industry, agriculture, medicine, biocontrol, etc. So all these microorganisms they are used in such areas for the benefits of humans. These organisms are used variously in food and feed technology. Various industries, apart from the food, various industries. In those industries, again, the microorganisms they are used. The waste utilization industry, energy, etc. Okay, so waste, that is sewage treatment, and all that we are going to discuss in brief in the next part of this session. Okay, so microbes in food preparation, the first and foremost very important part of using these microorganisms in the human welfare is the food preparation, how the food is prepared in the industries, in the food industries, and how these microorganisms they are playing a vital role in the food preparation that we are going to study in this part. Okay, so whenever the question is going to asked in the microbes in the food preparation, the thing you have to remember in this from this whatever you are studying from this microbial part, microbes in human welfare section, the name of microbe is very essential that you have to remember. Okay, there is no any shortcut. Okay, so you have to remember the name because this very short answer questions and number of multiple choice questions can be asked on this session. Okay, so the development of biotechnology occurred in two phases, that is traditional and modern. The traditional biotechnology is based on fermentation principle using fermenting bacteria. Traditional till 1971, whatever the fermentation techniques they were used. So in that part is called as traditional part. Okay, that was the old method where the principle was by using fermenting bacteria. So bacterial cells they were used to ferment that substrate or for the process of fermentation. That's it. These were used in the preparation of variety of indigenous fermented food products. Number of food products they were fermented, rather fermented food products they were prepared by this method. The example or the first part of this food preparation is dosa, dokla, idli. You all are very familiar with these terms and all these ingredients, right? Or these dishes, you can say. So the dosa, idli, dokla, they are fermented products, okay, and they produce due to activity of bacteria. If you have seen your mom keeping the whatever raw material she kept at night, so that if you have observed it, then you will understand what the fermentation process is, okay, because during the night time the bacterial cells, they ferment that material or that substrate, okay, and due to fermentation it releases carbon dioxide and it gives the puffiness. If you observe the dokla, that there it contains small pores, right? Idli, it is again very spongy, if it is very light, if it is not that stuff, okay, but it is very light and puffy, then we like to eat the idli, right? And again, talking about dosa, so dosa again, if you observe the preparation of dosa, it is very pepper like structure, it is very thin, but still it contains small bubbles or air packets, right? So all it is because of the process that is called as fermentation process. And they are fermented preparation of rice and black gram with the airborne leuconostoc and streptococcus species of bacteria. So as we were talking about the fermentation process, so your mom, she doesn't add anything right inside that, she doesn't inoculate anything in that, she just take a particular amount of rice, she takes a particular amount of black gram, then she keep that in the water, she soak the things, ingredients into the water, and what happens, the airborne leuconostoc, after mixing it, the airborne means the leuconostoc and streptococcus. Now we are talking about these two species of the bacteria, okay, and where they are present, they are airborne. Airborne means they are present in the atmosphere or in the air, okay. So as they are airborne, so they get mixed easily in that substratum or in that rice and black gram, and because of that, what happens, the natural fermentation takes place in those with those ingredients, and CO2 get produced during fermentation, causes puffing up of the dough. That is very important, okay. What happens because of the overnight fermentation process, because of the overnight fermentation process, CO2, that is carbon dioxide gas, get produced during fermentation, and because of the release of that carbon dioxide gas, what happens, that dough, it get puff, it get swells, it swells, okay. So microbes as the source of food, some microbes or their fruiting bodies are directly used as source of food, as they are rich in vitamins and proteins. So in the earlier example, that is in the idli, dosa, and in the dhokla, what we have studied, that the natural microorganisms, natural airborne microorganisms, they were naturally fermenting that rice and that black gram. But in case of these microbes, what happens, these are directly used as source of food, okay. The term SCP, that means single cell protein, it denotes dead and dried cells of microbes like bacteria, algae, molds, and yeasts. So what they gave, these are the things, okay, bacteria, algae, molds, and yeasts. What are they? They are single cell proteins, okay. They are taken, their intake is either in the form of dead or dried form, okay. Some mushrooms and truffles are directly used as food, so we enjoy the mushroom dishes, right? So mushroom is very healthy, it is it is a protein rich food, you can say. They belong to higher fungi, and they produce large fleshy fruiting bodies which are edible. Fruiting bodies are sugar free. That is again very important part, that why mushrooms are uh, mm, so likable, or why they are so favorite, why they are uh, eaten up all over the world, because fruiting bodies are sugar free, fat free, but they are rich in proteins, they are rich in vitamins, they are rich in minerals and amino acids. Now you can understand the importance of these proteins, vitamins, minerals and amino acids in our body. That is again very important, okay? And that's why the mushrooms, they are very famous. The food in the fruiting body is low calorie. That means it is low calorie food. That's why again it is used as food. Third part is dairy products. Now where how the microorganisms they are used in the dairy products, like lactic acid bacteria, that is LAB, like lactobacillus. Now this name you have to remember, okay? LAB is not lab. We call lab means laboratory, right? But this lab, this lab is different than the laboratory, okay? This is the short form of lactic acid bacteria. So you can get a short note on lab, so you have to remember these points, and you have to write about regarding this dairy products and all whatever you're going to study. Don't write anything regarding the science lab and all, okay? So lactic acid bacteria lab means they are lactobacillus. They are added to milk. Again, this is very important as you are talking about the dairy products, okay? So it ferments lactose sugar. If we add the lactobacillus into the milk, that means we are artificially inoculating the lactobacillus. Try to understand this, okay? In the dairy products, we are artificially adding something in the milk. It is not a natural phenomenon. It is not airborne microorganism, that you have to remember. But lactobacillus, it is inoculated, or it is added to milk, and in a certain amount. Again, that is again you have to specify. So it ferments the lactose sugar of milk. If the lactobacillus is added to the milk, then what process it occurs inside the milk? That the lactose sugar of milk gets converted into lactic acid. Now this is the first step. Then this lactic acid, when it gets formed inside the milk, then it causes coagulation and partial digestion of milk protein, that is named as casein. Okay, so there is partial digestion of this milk protein. That means there is something conversion which occurs inside the milk. So milk is changed into curd. So you get the difference between the milk. It is very its flow is different, right? It is thin, it is like just like the water. But curd, it is coagulated, it is somewhat thick, it contains some lump like structures, right? Precipitate like structure. When the milk it get precipitated, you can say when it's get coagulated, then it forms a curd, or the milk is changed into a curd. And yogurt and cheese. So because of the addition of this lactobacilli from milk, we get three kinds of products, that is curd, yogurt and cheese. The starter on or inoculum. Now starter means whatever you are adding inside the milk, that is called as starter, or that is called as inoculum. Used in the preparation of milk products, actually contains millions of lactic acid bacteria. Now what you have to understand in this, that the lactic acid bacilli, or these lactobacilli, they act as starter or inoculum, okay, for the formation of curd, yogurt and cheese. Now what is curd? Indian curd is prepared by inoculating milk with lactobacillus acidophilus. Lactobacillus acidophilus. Now, this name you have to remember if curd is asked. It also checks growth of disease causing microbes. Yogurt. It is produced by curdling milk with the help of streptococcus. Now, do you understand the names of microorganisms? They are changing. Lactobacillus, the name is same, but the next half part of the microorganism, the name is getting changing. Okay, why it is? Because we are changing the product which is getting formed from the milk. In the earlier example, the curd get formed when there is active microorganism named as lactobacillus acidophilus. In the yogurt, it is lactobacillus bulgaricus. Okay, so there is curdling milk with the help of streptococcus thermophilus. This again we require this one microorganism, and another is lactobacillus bulgaricus. Okay, this name you have to remember. Buttermilk. Now, what is how the buttermilk is get formed? The acidilated liquid left after churning of butter from the curd is called as buttermilk, which is again very healthy. The next is cheese. The milk is coagulated. The buttermilk, you can say it is charged, right? Again, cheese. The milk is coagulated with lactic acid bacteria and the curd formed is filtered to separate the whey. When the separation occurs, so the liquid gets separated from the solid part. Now the solid mass is then ripened with the growth of mold that develops flavor in it. So ripening because of the growth of mold develops flavor. So there is change in the taste. Different varieties of cheese are known by their characteristic texture, flavor, and taste, which are developed by different specific microbes. The Roquefort and Camembert cheese, they are ripened by blue-green molds, that is Penicillium roqueforti. Now these names you have to remember. And another is Penicillium camemberti. Camemberti. Okay, these are the two molds, name of the molds. The large holes in Swiss cheese are developed due to production of large amount of carbon dioxide by a bacterium known as Propionibacterium. This name you have to remember. If you are preparing for NEET examination, so this each and every detail is very important. Okay. So role of microbes in industrial production. Now, during fermentation, variety of products like alcoholic beverages, organic acids, vitamins, growth hormones, enzymes, antibiotics, etc., they are produced in the industrial production. So production on an industrial scale requires growing microbes in very large vessels. That these vessels they are called as fermenters. Fermentation process in the industrial scale, it is carried out in these vessels. So these vessels they are called as fermenters. The main function of a fermenter is to provide a controlled environment. Now, what is the function? Why the fermenter is used? Because it provides a controlled environment for growth of a microorganism or a defined mixture of the microorganism to obtain the desired product. That's why the fermenters they are used. Now, next, whatever part we are going to study in the industrial fermentation or industrial production process, that we will study in the next part. Up till now, what you have to remember is जितने भी माइक्रो ऑर्गेनिजम्स के नेम हमने यहाँ पे जितना भी आया है वो आपको स्टडी करना है, और जितना भी मैंने अलग अलग तरीके से इसको दिखाया है आपको. ओके, दीज आर काइंड ऑफ नोट्स. ओके, दीज आर द हाईलाइटिंग पॉइंट्स विच यू हैव टू रिमेंबर. इसी तरह से आप इस लेसन को अच्छे से याद कर सकते हो. ओके, एज फार एज जैसे मैंने पहले भी बताया इसका वेटेज कम है, पर नीट का अगर आप प्रिपरेशन कर रहे हो या फिर एमसीक्यूज में ये क्वेश्चन आता है या शॉर्ट आंसर क्वेश्चन में ये आता है, तो आपको ये याद रखना बहुत ज़रूरी है. ओके, तो आप इसको किस तरह से याद रखोगे? आपको इसको अलग से लिखना पड़ेगा। जैसे आप डेयरी प्रोडक्ट्स के बारे में लिख रहे हो, तो आप इन इंग्रेडिएंट्स का नाम लिखो, वहाँ पे इन स्पीशीज़ का नाम लिख दो, तो आपको ये याद हो जाएगा कि अगर ये है तो इसमें मुझे इन माइक्रो ऑर्गेनिजम्स को यूज़ करना है। अगर सोर्स ऑफ फूड आया, सिंगल सेल प्रोटीन के बारे में आप लिख रहे हो, तो आपको मशरूम्स या जितने भी सिंगल सेल प्रोटीन्स हैं वो आपको लिखने पड़ेंगे, उनके नाम। डेयरी प्रोडक्ट्स है तो उसमें लैक्टिक एसिड बैक्टीरिया के जितने नाम आते हैं, जैसे कर्ड, कर्ड आपने लिखा तो उसमें आपको अलग स्पीशीज डालना है, तो वो आपको याद रखना पड़ेगा। ओके, तो इस तरह से ये आपको याद रखना है, मतलब एक नोटबुक में आपको उसको लिख के रखना है। दैट इज इम्पॉर्टेंट। एक नोटबुक लो, उसमें इसका जितना भी इम्पोर्टेंट आपको लगता है कि इस पे ये क्वेश्चन आ सकता है, आप एक्सरसाइज भी देख सकते हो, तो आपको क्वेश्चंस भी पता चल जाएंगे कि कौन सा क्वेश्चन है या कौन सा आंसर उसमें आ सकता है ये, और उस हिसाब से आपको कौन से क्वेश्चंस उसमें आ सकते हैं। ओके, अभी जो रिसेंट 12th का एग्ज़ाम हुआ था, तो उससे उसमें डेयरी फार्म मैनेजमेंट और जो नेम ऑफ कैटल्स, काउज, बफेलोज वो वगैरह पूछा था, तो शायद से वो इस साल रिपीट नहीं होगा। तो आप उसको वंस या ट्वाइस आप उसको रीडिंग दो। बाकी का ये जितना भी पार्ट है जिस पे क्वेश्चंस नहीं आए हुए हैं, इसका मतलब बहुत ज़्यादा चांसेस है कि इस पे क्वेश्चंस आ सकते हैं। तो ये बहुत ज़रूरी है। कीप स्टडिंग एंड कीप आस्किंग क्वेश्चंस। ओके, आपके पास व्हाट्सएप नंबर है, यू कैन आस्क द क्वेश्चन। ओके, या फिर आप ये वीडियो देख रहे हो, तो आप उसके वहीं पे आप अगर आप क्वेश्चन डाल दोगे तो ये इजी हो जाएगा। ओके, जितने भी आपके डाउट्स है या आपको कुछ समझ में नहीं आ रहा है, या आप पढ़ाई कैसे करना है वो समझ में नहीं आ रहा है, या वॉट एवर योर प्रॉब्लम्स यू हैव, यू कैन जस्ट आस्क पर्सनली टू मी। ओके, दैट इज़ वेरी important right now. Hope you are studying and keep studying. Okay, so that's all for today's session. जितना भी नेक्स्ट पार्ट है इंडस्ट्रीज का, वो थोड़ा सा बड़ा सेशन है, तो वो हम एक साथ ही पढ़ेंगे और बहुत सारे इमेजेस के साथ हम उसके थ्रू जाएंगे, सो दैट विल बी वेरी इजी फॉर यू। सो हम बाद में मिलते हैं इन द नेक्स्ट सेशन। ओके, सो कीप स्टडिंग, keep preparing your notes, and keep asking doubts. So that's all for today's session. Thank you.